Take out the 8-channel comb and 96DW autoplate. Pipette 100% ethanol, isopropyl. Uncovering tinfoil covering on the DW autoplate. For convenience's sake, mark numbers for each column and row of 96DW plate. At 300 L isopropyl per well in columns 1 and 7 of 96DW autoplate. At 375 liters 100% ethanol per well in columns 3 and 9 of 96DW autoplate. In the biosafety cabinet, had 300 L samples per well in columns 1 and 7 of 96DW autoplate allowed samples to equilibrate to room temperature. Put the 96DW autoplate and the 8-channel combs into the instrument and run the program. After 7 minutes, remove the 96DW autoplate. Transfer the samples from column 6 and 12 to the centrifuge tubes and store them.